All right, everybody, welcome to the help video for our Building DNA Gizmo Lab. So this is the gizmo you should have opened on explorelearning.com. So if you haven't already done so, please launch this. You will also want to have open your Building DNA Gizmo Lab document. You can find this from your course content. You can click on the course plan for today, Thursday, January 9th, and it should be open on there as well. There's a Google Doc version or there is a Microsoft Word version. So we're going to use the Microsoft Word version, and we're also going to have our lab launched open. So that's right here, and I'm just going to go ahead and launch it and get started. So the first question on our lab document asked, it says, okay, pause before you start that, read your directions. So we're doing activity A, which is building a DNA molecule, and it says, if necessary, reset. So if you already had it open and you're pressing some buttons, just reset the gizmo just to go to your building process to start it. First question's asking us, where in the cell is this lab taking place? So if we go to our lab right here, what is this? So it's asking us, what is this? Where is this taking place? There are some hints over here you might want to read, and it says drag and join nucleosides and phosphates to build the left side of the molecule, etc., etc. And then it says, show the nucleus. Well, if you don't show the nucleus, guess what? There's nothing there. So I'm going to bet you money that this lab is taking place in the nucleus of the cell. So I'm going to go back over here, and that's what I'm going to write for number 1. Number 2, it says to follow the steps given in the gizmo on the left side to construct a molecule of DNA. So you're going to be actually constructing your own molecule of DNA. It tells you a hint for this model, for this gizmo. The DNA molecule is 2-dimensional and does not have the twist in it. So when DNA is actually being made, there is the twist, the double helix shape. So it'll be showing you that right there. So it will not be showing you the twist. It'll just be showing us 2-dimensional. So when it says build it, you're going to just drag something over. Boop. I'm going to drag that molecule over. See how I do this. Well, oh, that doesn't work. Why doesn't that work? Let's read our hint. Drag and join nucleosides and phosphates. Oh, so we need a phosphate. So let's throw that phosphate. Oh, there's a phosphate one on. Do I need another one? Oh, I do need another one. All right, so I'm going to keep building. Dun, dun, dun. Oh, got to drag another phosphate or it won't let me drag it. Keep building, keep building, keep building. Go ahead and keep building yours, and then we'll start here in a second. Pause the video if you need to. All right, I just went ahead and paused my video. I've built my whole left side of my DNA molecule. I just dragged the phosphates, dragged the nucleosides over. So I'm going to go to my lab document and fill in my chart now with my left side. I just went in order of the gizmo, so it was super easy. So it was just A, A, C, C, G, G, T, T. So that way whenever I write it in here, super easy. You just write A, C, C, G, G, and T, and T. Okay, so you've got your left side built. And then it's going to ask you right here, what is supposed to go on the right? So if you were in class on Monday, you probably know the answer to this, but you have base pairing rules. A always pairs up with T, C always pairs up with G, G with C, and T with A, vice versa. So if you go to your gizmo and you're building it, if you didn't know that, you could try to build it, and you could see, like if I pair A with A, boop, it doesn't let me do it. If I pair A with C, nope, doesn't let me do it. If I pair with G, nope, doesn't let me do it. So I have to then realize, oh, T is the only one that's going to work with. And for this one, you also have to make sure you have those phosphates on there before you can build your next strand. So you do have to have those in order. So now it's going to go the opposite of what I just did. So I'm just going to keep building this and keep building it until I have a completed DNA molecule. Both sides, left and right. So pause the video and make sure yours is completed. Okay, so as you can see, I've got mine now all completely built. However, it doesn't have anything else for me to do. So something's not right. So you'll notice you should still have a phosphate over here. Or if you still have a phosphate, you need to figure out where it goes. So I'm going to try to put it on the top. As it tells you right here, if you put it in the wrong spot, it's going to tell you why it doesn't let you do that. It says lagging strand does not end with a phosphate. Do not add a phosphate to the top of this strand. So since this is my first strand that I built, that's called the leading strand of your DNA molecule. And this over here is the lagging strand. So then the next step means you must put this one at the bottom. So since that's our lagging strand, it's going to go at the bottom. Oh, and now that I've done it, we have our new step. So I'm going to go ahead, once it's all built, you want to make sure that gets copied over into your question for the right side over here. So you're just going to go ahead and put all your base pairs that matched up with it in your chart over here, so on and so forth. Okay? So that should be completely done. Now you're down to number 3. So it's asking you right here, what makes up the sides of your DNA molecule? What are the blue molecules? So look at the sides of it. What are those blue things called? You guys just had them in a chart over here. If you're not sure, go back and go find those answers. Okay. B, what makes up the rungs of the DNA molecule? The rungs are these letters. So what are those letters called? The rungs are like the steps you would step on if that was a ladder. So you want to figure out what the names of those are, and that's what you would write down for B. Number 4. This is just your answers that you already know. What are the base pairing rules? What does A always join to? And for simplicity, you can just write the letter. So we know A always joins with T. Complete it for the next 3 as well. I'm not going to give all the answers in this video, just a couple of hints to get you started. Okay? Once you've got that completed, you're going to scroll down and you're going to do a little practice. This is not in the gizmo. This is just practice on your lab. So you're going to fill this in and tell me what the base pairs should be matching up to. So if this was your strand of DNA, what would the right side be? You just fill in those letters. Super duper easy. Okay. Activity B is starting with DNA replication. You have to make sure the hint is still on where we ended. The DNA molecule is complete. If it's not there, if you reset it for some reason, you have to rebuild your DNA. Okay? So if you are not there, reset and build a new DNA molecule. And then once you're there, that's where this lab starts, this activity. So the first one's asking about an enzyme called DNA helicase. So over here, this is our enzyme called DNA helicase. I just realized I could have made this full screen the whole time. So as you're working through your lab, the first step said to release this enzyme and watch what happens. That's your answer to activity B number 1. So I'm going to release it and let's see what happens. Okay, so our enzyme helicase, which was the blue little enzyme helper guy, he came in and kind of unzipped the strand of DNA. So that's what you're going to write for number 1. Now, you should notice there is another enzyme that has shown up. Now, instead of helicase, we have an enzyme called DNA polymerase. So DNA polymerase, we're going to actually figure out what it does next. It tells us before we can do that, it says now that you've got this polymerase there, you'll notice that on the right-hand side, after you release it, there are, let me click it to release it. There are 2 groups over here. There's nucleotides that are left facing, and then there's nucleotides that are right facing. So these are the ones you'd put on the left, these are the ones you'd put on the right. Okay, so remember this process that we're looking at right now is DNA replication. The word replication means we are going to copy the DNA. So we've unzipped it, and now this DNA polymerase is going to be our little helper protein that's going to come in and partner up all of our enzymes. So you'll see when I drag in A, it kind of comes along and it's our helper. It's guiding it to where it needs to be. Now, if you put it in the wrong spot, so A with A, boop, polymerase says, no way, Jose does not bond with it. So it lets it go back to the building area. However, if you pair it with the right one, it says, yay, and it lets it stay. Okay, you can pair your polymerase, you can pair your nucleotides up in any order. So you don't have to put them in order when you're rebuilding DNA. When you're replicating DNA, you don't have to put them in any particular order. You can put them wherever you want. Okay, so we're just going to keep going, plug these in quick, get it done, and when you're finished, you will end up with 2 identical strands of DNA. So why don't you build yours, see what your results look like, and then you should have an answer for numbers 2 and number 3. Okay, I just went ahead and built all of my DNA, the replicated DNA strands. So remember, each side of this was the original DNA, and now it's been replicated and new bases have come in and been paired up. And if you look, they're exactly the same. Even if you built yours in a different order than I built mine, so remember, it didn't matter when you built them. As long as you have them all paired up, you'll notice the strand on the left is the same as the strand on the right-hand side. So that should be a helpful hint for number 3. Number 4 is asking you, why is DNA replication such an important process? So we talked about this part in Class Connect. For this, the reason it is so important is without DNA is our instruction manual, basically. It has all the genetic coding for every cell in living organisms. So if we don't have DNA and cells are reproduced or they're divided during cell division, there won't be any instructions, and those cells will be kind of just hanging out without guidelines of what they're supposed to be doing. So DNA replication is really important. It has to happen before cells replicate. Okay? So something to write down there. Make sure you write it in your own words. Number 5, it's asking you about errors called mutations. So it says sometimes these occur during DNA replication. What are some of the possible consequences? So what I mean by that is if our DNA polymerase did not come over and instead of this A right here, I'm going to keep that arrow there so you guys can just see it. Instead of that A there, a T paired up with a T. Well, this is not proper base pair rules, and that means something is going to happen with this DNA strand. This segment of DNA might not function properly. Maybe something extra will be coded for. It could lead to the growth of something extra as well. Maybe something along the lines of cancer. It could lead to a genetic mutation. It could lead to tons of different possibilities. There are good things that could happen as a result of it. Like, for example, my red hair is a mutation. But there are also some bad things that could happen. And sometimes when there's a mutation, there's no issue. There's no actual physical issues that come as a result of those genetic mutations or those DNA replication errors. So lots of things could happen. I just want you to pick and choose one and explain it. So that's why the question is asking. All right, that's easy. 11-minute help video. Hopefully you guys got through it. If you need to, go back and rewatch it. Pause it as you do it. However you choose. Please get your labs done and turn it to the Dropbox. Have a great rest of your first week. Bye!